Hi, my name is Inaya. Our project aims to study the relationship between retinal transplant organization and visual acuity of rats through the use of optokinetic testing, light and dark box testing, and histology samples stained with hematoxylin and eosin, also known as H&E. By identifying how the structure of transplanted retinal tissue influences visual acuity, we aim to understand successful transplants and how they can be improved as treatment for retinal degeneration. Each member of our team brings a different source of inspiration leading up to the formation of this topic. I was drawn by the intersection of engineering and medicine, especially how data and experimental design can translate into healthcare solutions. Through my work with retinal stem cell research, I've developed a strong understanding of how to collect and interpret data in a way that helps make sense of vision loss and supports progress towards improving treatments. And next, introducing the other members of the team.
Hi, my name's Jenny, and I was driven by an interest in how biological structures relate to the function and motivated to gain hands-on experience that connects research to future clinical applications. Working with retinal transplantation made this more real for me, since I could see how small structure changes that affect visual outcomes.
My name is Emilia, and I'm inspired by regenerative medicine and the potential of stem cells to create more patient-specific and sustainable treatments. This particular project is important to me, as by narrowing down the retinal factors that impact vision, we can improve the precision and effectiveness of these treatments.
My name is Anya Thakar. As an applied physics student, I am driven by the intersection of quantitative analysis and clinical application in the biomedical field. I was drawn from physics research into the medical sphere because of the immediate impact of research on patient health.
Overall, in this study, we focus on how well retinal layers are aligned and their thickness in the H&E samples, specifically the photoreceptive layer. Then this is checked through measuring visual acuity through behavioral tests, which is then compared to histological samples. This connection allows to see the relationship between the two and how well rats respond to visual stimuli and whether the transplants worked.
Together, these perspectives shaped a collaborative approach grounded in curiosity and innovation for this project of how we hope to contribute to a deeper understanding of how stem cell-based retinal therapy can restore vision. We believe that small improvements in consistency and effectiveness can have meaningful implications for treating degenerative eye diseases. Overall, this experience further strengthens our skills in experimental design, data analysis, and teamwork, further inspiring us to grow as future scientists and healthcare professionals.